All right, part 2 of our protein synthesis, which is really step 3 of our central dogma that's known as translation. So I'm going to talk about how we convert that newly made messenger RNA transcript into protein, our very special molecules that we need to survive, right? So I'll hide myself over here. As we talked about before, we know that there are many different types of RNA out there, and those are going to be temporary molecules, um, short-lived. They utilize the uracil in them. They have a ribose sugar in them that's different, and they're single-stranded. But it's genetic materials. It's instructions for that organism, but um, it's not like the main player of our genetic material like DNA is. So there's different types. We've talked about messenger RNA, where it's going to be moving the genetic material and that code so that it can leave the nucleus. um, there is tRNA, which is transfer RNA, like I talked about before. It's oftentimes takes the shape of a T when it's folded in its 3D pattern. Let me pull that up here so I have my laser. There we go. Here's my tRNA with its shape, right? And that's going to be transferring amino acids in order for the polypeptide sequences. And then our last one is going to be rRNA, which is ribosomal RNA, which is going to make up the ribosome units that we see during translation. So there's some visuals for you as we're working through. So translation is going to be RNA-directed synthesis, RNA-directed creation of proteins, and it's going to utilize all of those RNA components as we're working through. So we're going to be using that messenger RNA, that newly made messenger RNA transcript from transcription. It's always going to be read 3 bases at a time, and that's called a codon, right? A segment of 3 messenger RNA nucleotides. And that's going to give us the code specific for the amino acids that go along with it. So AUG is a start codon that we're always going to have on our messenger RNA near the front, near the cap of that messenger RNA. It'll go AUG because that signals, hey, let's start translation here. So anyway, that one is associated with the amino acid methionine. So anytime the codon AUG is read, it's going to draw in a tRNA anticodon molecule, which is going to bring along its buddy methionine with it. Okay, so tRNA is going to have an anticodon on it, which is a 3-base nucleotide combination, which is complementary to the messenger RNA. And it's RNA, so it's still going to have A's, U's, C's, and G's, just the opposite code of what we see. So here's our codon and anticodon that are matched up with each other. That transfer RNA links momentarily to here's my codon down here for messenger RNA, AUG, and then my transfer RNA anticodon comes and links up. It's confirmed that they are matching complementary bases with each other, and then from there, tRNA is like, hey, methionine, come on over, and methionine travels and is attached, at the top, and we'll start adding along with the rest of the amino acid sequences that occur. So that is kind of the differences between our messenger RNA and our tRNA, both using RNA nucleotides, both utilizing the 3-base readings frames, but we got codons versus anticodons, and transferring amino acids versus not. So sorry, I zipped through that. Just like transcription, there's 3 phases, and they're both called initiation, elongation, and termination for both transcription and translation. They're just going to be doing slightly different pieces. So again, we want to be able to translate and create protein when we want it, however much we want it, and at the right times, and not just constantly making proteins for no good reason, because it takes energy to do that, which is very valuable. Um, so there's got to be an initiation sequence that is read in order for this to happen, which I already mentioned as well. So our ribosome is going to be hanging out there. Messenger RNA is going to come and attach to that ribosome, and it's going to read the initiation sequence of AUG. And it's going to say, hey, let's start translation. So once it reaches that AUG codon, it's going to draw in some initiation factors. Which are a lot like the transcription factors that we saw before. So these globular, again, proteins are going to come and attach to the messenger RNA and the ribosome area, and just kind of prep it and make sure everything is locked and loaded in place. And that way the messenger RNA isn't just going to like flip out of the ribosome and then we have some issues. So it's going to hold everything in place and allow the processing to begin. All right, so there are actually 3 sites on our ribosome that are going to be binding sites for our tRNA molecules. So here's my messenger RNA down here. We can see AUG was red, so it's going to start. There are 3 sites, the E site, the P site, and the A site. Okay, APES, if you're working backwards, Environmental Protection Agency, EPA, if you want to work that direction of how you remember it. But the one in the center is our no, I'll start backwards. Sorry. I'll start with the A site. So the A site stands for attachment site. The codon is red, and it needs to find its complementary anticodon on its tRNA molecule. So it'll read that 3 base UUC and say, okay, the next tRNA molecule we need is AAG, come on down, right? It's going to call to it, and that tRNA molecule is going to come over and just kind of attach lightly and bind into place, right? When it's its turn, it'll be shifted over into the P site. And the P site is going to be named the P site for the growing polypeptide chain that occurs. So it locks and loads here with the anticodon and the codon. It's confirmed that they are a match. That tRNA molecule is going to drag in its specific amino acid at the P site and bond it with the growing polypeptide chain. So these are peptide bonds that are occurring between them. And then once it's confirmed and it's attached and everything's good, it's going to move to the E site, which is known as the exit site. And that tRNA molecule leaves and floats back out there to go find its specific amino acid so that it can come back and work through if it's needed again later. So it just kind of shifts through the attachment point to the polypeptide point, to the exiting, and that growing polypeptide chain up here is just going to keep on going until it reaches that last termination sequence. So let's keep going. You can kind of see, again, I'm trying to get myself out of the way so we get this visual. Here's the small ribosomal subunit where the messenger RNA is attached, right? And then the large ribosomal subunit has the APE sites on it. We can see the codon and the anticodon over here, how they're matching up with their complementary bases, right? And once they're matched up properly and locked into the P site, that's when the amino acid is going to come and join in. And we can see like this complex and this transformation process of going from the A to the P to the E site. It's utilizing some energy sources like ATP, but it's kind of like a cousin of it, GTP. Anyway, so that's how it moves it along. And then it'll keep on going through those 3 subunit codons and shifting it through kind of like a typewriter until it encounters the stop codon. So that was phase 2, elongation, when the amino acids are added one by one next to their previous amino acids. There are different factors that can come and attach to it. So again, when the codon of the messenger RNA is paired with the tRNA anticodon, there has to be that recognition and confirmation happening. There has to be peptide bonds forming when those amino acids are linking with each other up towards the top. And then translocation, it is that term refers to when the tRNA molecule is moving from one spot to the next. So those are our elongation factors that occurs. So here's our codon recognition. This is technically translocation when it's moving along, and then we can see the polypeptide, the peptide bonds being formed over here. And I know that's a blurry picture, but gives us our visual of it. And then it's going to hit the termination sequence at the end. There's actually 3 different termination sequences on messenger RNA. Just to kind of give some unique factors to it. But once it reads that messenger RNA codon of termination, there's going to be a release factor that comes into the A site of our ribosome, then attaches there and says, hey, we're done. Everything is going to like separate off of each other from here. And so it's actually like a hydrolysis reaction where water will come in as well with that release factor, and it's basically going to cause like everything to just explode. So that messenger RNA that's temporary, if it doesn't need to repeat and make another identical protein sequence, that messenger RNA, all those nucleotides are going to break apart and go back into the free floating cytoplasm. The subunit for the ribosome can separate. The growing polypeptide chain is going to be released from all of that so that it's just there free floating and it can be folded into its proper 3D pattern and shipped off to where it needs to go. So the termination point brings in release factors and lets everything separate from each other. So here's what those release factors might look like. It's kind of like a blockade where it's going to shut down any more tRNA molecules from attaching over here. And here are those 3 different stop codons, but you don't have to memorize them. So it blocks it, and that's going to cause some hydrolysis where it's going to detach the tRNA molecule and get those out of there. We got our ribosomes separating over here. We have our free polypeptide chain, aka protein, that's going to get folded up over here. And eventually this strand of messenger RNA is also going to get broken down. But it'll hang out longer if it is needed in greater quantities. So again, this is going to talk about coiling, winding, going through our quaternary process of folding and creating our 3D shape for our protein. Sometimes they have some post-translational modification, like as the Golgi body. Okay, we're getting back to that thing. As the Golgi body is shipping it out, sometimes it does some additional modifications. So that might happen. Some of those polypeptides are ready to go, but as soon as they get to the site that they're needed, there could be an enzyme that chops off a piece of it, which actually activates that polypeptide or that protein that was just made. So that's what the second bullet point is referring to. And then sometimes those polypeptide chains that were just folded into 3D shapes need other polypeptide 3D shapes. So they'll kind of clump together and wait for all their other 3D protein buddies to join and create this globular protein, which I've been talking about before. So again, here's the reminder of that protein folding. We just made our growing polypeptide chain through translation. Then it can go through either the alpha helical spiraling or the bead pleated sheet where you're like folding paper in half, or more than likely it's a combination of those two, right? Tertiary structure is when some side chains are going to be bonded together with those kind of temporary bonds. And then our quaternary one is our 3D version. Furthermore, talking about mutations, right? So we have polymerase molecules that'll go through and check and do some excision repair if there's any issues with base pairing that went on, um, to try and correct everything. But there are still some bases, 1 in a billion bases, that get past our polymerase molecules, our checks and balances piece, that leads to some mutations. So I already had you look at a few of them, but I'm going to give you the details of mutations here. So a point mutation, point is one base change that occurs that was not corrected. So just one base pair. So maybe an A was paired with a G. Oops, that's incorrect, but it wasn't caught. That single nucleotide base is called a point mutation. Okay. It could be a switch, like I said, an A paired with a G, or it could be where one base was added in where it wasn't even supposed to be, or one base was removed or taken out of that sequence. So additions, deletions, or single nucleotide switches can be subcategories of point mutations because one thing was changed. I'll keep going. For those point mutations, they can be known as silent mutations if it is a single nucleotide switch. An A is paired with a G when it should not be. Most of the time, those single base substitutions are what are known as silent mutations, where they actually don't have an effect on that polypeptide amino acid chain that eventually gets made. Um, so those amino acids that are brought in, it's still the correct one, and there's no issue going on. Because there are 20 different amino acids out there. They have a whole range of codes that are associated to them. And so it's possible that you still get the right thing, um, which is really, really cool that we can still have some errors, which make us unique, but not have an effect on our proteins. But if there is something that goes through and causes an incorrect amino acid to be coded for, that is called a missense mutation. Um, so when an A pairs with a G instead, and it causes the wrong amino acid to attach, that's called a missense mutation. And then a nonsense mutation is like when a single base or multiple bases have been added or deleted from that growing chain, and it just completely messes up the whole frame shift reading. Sorry, it messes up the entire reading frame of that messenger RNA. So as it's read codon by codon, instead of it saying A, C, G, it says A, C, C, and then the G occurs. It's going to cause this whole entire shift with every other codon after it, which usually leads to a non-functional protein or some incorrect proteins. It may never encounter a stop codon as well. So that's called a nonsense mutation. So there are some examples of that here. Silent, where it has no effect on the proteins chain. And we're going to focus over here where it goes GTA. And this one has GTT. They both code still for valine, the amino acid valine, so we don't have an issue. So that would be a silent mutation. If there is a base substitution that causes an issue, like this one where we have CCC as the original one, but then we have ACC, that originally coded for proline, and this new one codes for threonine. That's going to cause the wrong protein sequence, and cause an issue. Nonsense is where everything gets all mumbled jumbled up, right? So in this case, TAC, we're coding for tyrosine, but instead it writes TAG, and that codes for a stop codon, which is AUC, by the way. in messenger RNA speak. It'll code for an early stop codon in our amino acids. And then what we originally were supposed to have, which is somewhere between 20 or 200 amino acids long, now is only 4 amino acids long, which would make no sense. And then frame shift mutations are the really detrimental ones, the ones where all of the genetic material gets messed up because of an insertion or a deletion or an early stop codon. So if we look at this over here, 2 bases were added in. So two bases were deleted. The original sequence says GTACCC, and now it says G. It lost the T and the A, and these C's shifted upwards. So that's why it's called a frame shift, because those bases are moving and shifting locations. So now we still have valine as our second amino acid, but then everything after it is no longer the same. And so the whole entire rest of that code is messed up. And so that's a frame shift mutation. I'm going to stop there.